But not when you hold me. Your beauty weighs on me. This feeling's too good.
Hey, listen.
Lonely,
Hey, listen.
but not when you hold me. But not when you. But not when you hold me. The beauty lonely. Philo. But not when you hold me. But not when you. But not when you hold me.
Hey, listen.
Lonely. But not when you hold me. But not when you lonely.
Okay. Welcome everyone to today's part of the proteins and protein folding workshop. Today is, I think, going to be very interesting. It's going to be a little bit different from what we did the previous times. We're going to start, of course, with a little bit review of what we did last time, so everybody knows where we're at and has the same understanding a little bit that we need to continue this workshop. Again, I would like to hear feedback. And, oh, I see, sorry. I think the feed, that my voice is double comes from Zoom. Let me check. Okay, right. Okay, yeah, somebody muted me. So let's make sure that everybody knows what we're talking about, and then we're going to continue into a new topic today. So first, last time, mainly what we talked about was the folding process of proteins. So we talked about how something comes from its peptide, unfolded, unstructured state into its what we call the native state, right? The folded and the functional state that a protein can be in. Right? So we even made our own protein or our own peptide on PyMol, and we kind of scrunch it together and randomize it to see that there are many different variations and many different ways a peptide can fold. But one thing that is true for all of them, that in the folding process, the energy of a protein is always decreasing, and the protein basically wants to get to a state that is very stable, right? Has a low energy amount. So this can be a native state, but we also looked at that this can also be a non-functional state, right? Either a amorphous aggregate or, for instance, amyloid fibrils, right? Those are states that basically the cell or the machinery that is producing proteins wants to prevent. And we also talked a little bit about proteins that do that. We talked about the chaperones, right? So if a protein is or during its folding process, chaperones basically can make sure that proteins fold themselves into the correct shape rather than into the non-functional state. Right? They do that with different processes by just binding with them, by modifying them, by basically we talked about GRO-L, GRO-S, which is basically chewing them in a way to put them into the correct state. But there are many different ways they do that. Okay. And okay, that's actually not about chaperones, sorry, the title is wrong. In addition, I wanted to add something a little bit because I thought it would be interesting for you guys to know. Maybe, I don't know who of you maybe thought about why are we using ethanol, right? So always when we want to disinfect things, for instance now with the coronavirus, we're trying to disinfect things with ethanol and even washing our hands with ethanol. Why are we actually doing that? So this picture is supposed to basically show you what ethanol does. In the previous workshops, we talked a little bit about why certain structures are formed in proteins, and a lot of that depends on the environment the protein is in, right? So in the normal hydrophilic environment with water, right, there are tertiary structures that are performing hydrogen bonds, right? Hydrogen bonds basically make a protein stable, give it a lower energy, and it forms different structures that are very favorable with a lot of hydrogen bonds, for instance, like an alpha helix or a beta sheet that we talked about. And here's, for instance, one of those hydrogen bonds that can happen, right, between asparagine and tyrosine, right? So you see the hydrogen bond is between this H and this O. And in a 3D structure, you would see down here, it's maybe a little bit low resolution, but I hope you can see it. Right, so this is what would happen with water. But as soon as you add ethanol, this hydrogen bond is replaced by actually ethanol. Like, this is the formula of ethanol, and this forms this hydrogen bond instead of the tyrosine. They each get their own ethanol molecule that binds to them, and what would happen in the 3D structure, they would basically kind of move apart, right? They would not be connected anymore. So what alcohol does to proteins, it changes their shape and makes them unfunctional, right? So the reason why we use ethanol to disinfect our hands is because the proteins of the virus, they, like basically most other proteins, they are sensitive to ethanol, and they change their shape and they don't fold back correctly, even if you replace it with water again, and they stay in this unfunctional state instead of the native state. Right? So basically you've killed the virus that way. Okay, so this is how basically a real life application of this folding process that we basically do nowadays almost every day. All right, so also something that we talked about last time was a software called Folded, where you can, with like a puzzle game, you can find out and test out different foldings or structures of proteins. Not with a computer like we talked about. You can also kind of predict them with computers, but basically the best computer so far is the human brain. So we can kind of figure out, oh, this amino acid could fit here, because we have kind of way of logical thinking. Our brain is able to combine a lot of variables together to then form a structure, maybe a good answer to many of the folding questions that we have when it comes to proteins and how they get the shape that they have. And yeah, this kind of puzzle game, I told you, has actually also, it's not only a game, it is real research. And one of these examples, how this game actually contributes to research, I wanted to show you in this little video. And yeah, let's watch it together.
Welcome to a very special update from Folded. In this video, we're taking you inside the lab here at the University of Washington Institute for Protein Design. We're recording this video in the midst of the global coronavirus pandemic. Most of our team is working from home at the moment, but some scientists are still coming in because they are working on a possible coronavirus cure. All the experiments you're about to see are the same ones that we will be conducting soon on some of the very best Folded player designs. If you aren't familiar with Folded's coronavirus puzzles, we've got a video on that in the description below. Right now, the team has identified 99 of the most promising Folded player solutions. We'll begin testing those molecules shortly, but we didn't want to wait to show you what that's going to look like. The goal of these experiments is to manufacture proteins that were designed on a computer, then to test them to see if they bind to proteins on the coronavirus. If they do, these molecules might be considered candidate antiviral drugs for blocking COVID-19. We want to emphasize that what you'll be seeing in this video is just the first step of our drug development process. Much more work, including clinical trials in humans, will need to be done before any new protein drug is ready. Those crucial experiments will take time. The scientist conducting these early experiments today is Longxing Zhao, a postdoctoral scholar here at the University of Washington. He's working to stop COVID-19, but before you get worried about his health and lack of face shield or respirator, there is no live virus in our lab. Instead, we use this. This tube contains a purified sample of the novel coronavirus's spike protein, which is the same gray protein you've seen in all folded coronavirus puzzles so far. Because we work with a recombinant protein, there is no risk of infection. Our scientists have been designing proteins against this target, just like you can do in Foldit. Because they work here full-time, they've had a head start. Our experiments always begin with synthetic DNA. DNA is the blueprint for making proteins. Thanks to new DNA printing breakthroughs, it's now possible to create from scratch thousands of synthetic genes that encode whatever proteins you want. This tube alone contains over 12,000 genes, and it costs about as much as a used car. Once we've acquired the DNA that encodes the new proteins, we need to put it into cells so that they can carry out those genetic instructions. In these experiments, we grow yeast cells. Laboratory yeast are great at making proteins. In this case, we've genetically engineered each yeast cell to not only make one specific designer protein, but to also decorate the outside of the cell with many copies of that protein. This is called yeast display. For a designed protein to block coronavirus, it has to bind to it. We measure whether this binding is happening using an instrument called a flow cytometer. This machine flows a thin stream of liquid containing the cells in front of a series of lasers. The stream is so small that each cell passes the lasers one at a time. The lasers illuminate fluorescent molecules that have been attached to the proteins. The spike protein fluoresces at a different wavelength than the binder. If the binder actually sticks to the spike, the machine will report seeing both wavelengths at the same time. That's how we detect binding. Today, we found some hits. These molecules will still need to be studied more here at the University of Washington. They may even need to be further optimized on the computer, including inside Foldit. So stay tuned for more puzzles and more updates from the lab. As always, thanks for watching, and thanks for playing Foldit.
Okay, so I thought that was a very neat video that shows basically how things that you could do in Foldit find real-life application, for instance now fighting the pandemic. So if you have time, and if you're bored at home, and if you want to learn and basically play games at the same time, check out the program that I showed you last time, Foldit, right? It's definitely worth a look. Alright, and with that, I would like to go into basically now the topic what proteins can do. So until now we talked about the structure of proteins and how the structure is like develops or is basically formed. But now we want to take a look at what actually proteins do, right? And I have another short video. I mean, it's a little bit longer, but I'm gonna skip through it. That just shows you what is happening in the cell. This, of course, is by far not everything that is happening, but something that I always felt for myself is very interesting. When you try to imagine, not only from the pictures that you have from biology or from a book, but also rendered in 3D, all the processes and all the crazy things that are happening inside cells. So, So just take a quick look. Okay. I don't know, I don't think we need the sound, but yeah. So, for instance, here you can see the red ribbon is DNA, for instance. And what you see in purple is called a histone. At the same time, for instance, the green parts that are moving here are transcribing the DNA into mRNA. Those are proteins that then transport the mRNA outside of the nucleus through nuclear pore complexes. As an example. Yeah, more histones. Maybe histones you have heard before because they are responsible for DNA being able to assemble into chromosomes, right? Those X-shaped things that we have 24 different types of, that encode all the genes that are in the cell, right? So they form bigger and bigger and bigger coils. And then this is what you would see under a microscope. During mitosis. Then a very interesting protein, for instance, that I like personally is DNA polymerase. So this is basically what you're seeing here. This crazy machine is active in every cell at any time point and is basically copying DNA. It's just making a exact copy of the original DNA before basically cell division. So I think this especially is a good example for why we call enzymes molecular machines. But because this definitely looks like maybe what you would find in the factory producing different products. Those are transcription factors. We don't have to go into detail about those too much. Yeah, this is transcription of DNA into RNA. Maybe here a little bit more in detail. There's a site that basically unwinds the DNA, and then the nucleotides of the RNA, they try to fit exactly into these pockets. But of course, that's happening at a very, very fast rate. And so on. Let's see, is there anything else? Okay, maybe that's okay for now. I just wanted to give you an idea of what proteins and their function look like in a cell, basically, because so far all we looked at is basically still pictures. But the truth is all molecules are in movement, right? So this, I don't know, I've heard from some people that they don't like it because it looks kind of gross. I don't know, but I think there's a lot of fascinating things that you can find in those natural processes, because they're from evolution, they have been perfected to a degree which is incredible. So in the coming workshops, we will be looking at different types of proteins. I guess it's more like a personal selection of mine. It's not, we of course can't look at all proteins. So we're going to look at proteins that I think are very, very fascinating and have very interesting mechanisms to look at and how they work. And today we are going to start with motorproteins. So just as a rough outline, so today I'm not sure we're going to finish motorproteins because there are different types and all of them are very, very interesting. But after proteins, we're going to look at transporters. After those, we're going to look at receptors, and at the end, we're going to look at enzymes. Yeah, so probably each of these topics can take up more than one single workshop, so that might take a while to get through all these topics, but it's all very, very interesting. Okay, so motorproteins is the topic for today. So, I mean, the name of them is very kind of logical if you think about how they work. So, first of all, I would like to have you think about what does a motor actually do and how could it relate to what the protein does. Right? So, motors in real life I hope some of you have seen this kind of mechanism before. Basically, they are in every car or generator, and what they do is they use gasoline. Gasoline is then moved in a combustion chamber, is ignited, turns into gas with a small controlled explosion, and this gas and the expansion from this, when this energy release, then pushes this piston down, which turns the crankshaft, which then turns the wheels. Right, and the rest, where basically the fume is released through the exhaust. Right. So this is how a motor works. And if you think about the concept, right, so a motor turns gasoline and oxygen into CO2 and water, right? The energy that is released in this process in the combustion engine is then turned into movement, right? So in a way, you turn chemical energy into kinetic energy. Well, kinetic energy is just another fancy word for movement energy. Okay, so if you were trying to transfer this process into a living being, right? Actually, something very, very similar is happening in humans and animals and all living things. Right? So in living things, sugar and oxygen are turned into CO2, H2O, and ATP. Right? So because we have to eat food, we breathe oxygen and we exhale CO2. And water is produced also heat. And for us, mainly what's interesting is ATP. ATP is basically the energy currency of the cell we can use for many types of processes. And today we're going to look at how this energy generation process, which results in a lot of ATP being produced, is turned into movement, right? Right? And turning ATP means chemical energy into kinetic energy, the movement, can, for instance, be seen in muscles. But we're also going to look at some other types of proteins found in different parts of the cell. So, and all of them are called motor proteins, right? To be exact, not all of them have to use ATP. There are also other forms of chemical energy that they can use. But today we're going to mainly focus on the ones that use ATP. And we're going to start with muscles, right? So everybody knows what a muscle is. Everybody has done sports before, but I don't think many of you actually know what muscles do. And today we're going to go into the very, very detail of how muscles work. All right. So going through this this picture of muscles, right? So you have, for instance, the muscles in your arm, the biceps here in this case, which is formed of muscle bundles, right? So you have muscle bundles and you have veins and arteries in between that are there for the blood flow and the distribution of energy and oxygen. These muscle bundles then are made up of different fascicles. And fascicles, I mean, what you can see here as a purple, are basically, they're made up of long cells, right? So, and the purple thing is the nucleus of the cell. And inside these cells, you can find muscle fibers. And in these fibers, you can find different fibrils. And then the other part is called the sarcomere. Sarcomere? I'm not exactly sure. And what they're basically made up of is actin and myosin. So those are the main parts we're going to look at today, right? So from this structure, I mean, how does this turn into movement, right? And when all you need is basically actin and myosin as a protein. There are also some other things that I would like to mention just very shortly. There's like a Z line, where it's basically is like an anchor for the beta actin that's reaching inside. And I think the space in the middle is called the M line. Yeah. Okay. But what is mainly interesting for us today will be the myosin. So, and the myosin in this illustration is made up of a alpha helical coil. We've talked about that, I think, 2 workshops ago, right? So 2 alpha helices that are basically twisted with each other. And then you have 2 light chains and a motor domain. Today we're going to basically be mainly interested in this motor domain. And here it would look like, here's what it would look like in the muscle. So basically you have also many twisted strands of one myosin molecule, which form like, I don't know, it looks a little bit like a twig or something, right? So with many different motor domains sticking out in the side of it. Okay, so how does it work? So before we go into the detail, I just wanted to show you in general with this animation how the movement is done, right? So, and don't worry if you don't understand it completely in the beginning, but we will go over it in detail slowly. Right? So here we can see the motor domain, right? And the movement that is mainly happening is basically this yellow part. So what's happening, I hope you can see it well. There's something basically exiting and entering, right? This exiting and entering molecule is ATP, right? ATP goes in, is split up, the energy is released, and then it goes out. In detail, so basically when ATP goes in, this motor domain releases itself from the actin filament. It changes its shape and stretches out. Right? Let's wait one more time. So it stretches out. Then the ATP is split into ADP and P to release the energy. This causes the motor protein to attach to the actin again. And when it's attached, it's basically like a wound up spring. It releases the energy that it stored by stretching out to then move the actin filament backwards. Right? So again, I think the analogy of the spring is very important here. Right? So the spring is loaded, right, by actually binding ATP. So the most energy that this conformation has is in the stretched out state. And once it binds, it basically tries to release the energy by going back to the low energy state, right? Which is in the contracted state. Right. So then the ADP and P goes out, ATP goes in, it releases itself again, and then the whole cycle is continued. Right? Okay. If there are any questions about that, feel free to ask in the chat, because I'm sure there will be. Okay, so what I want to do with you now is take a better look, right? So similar to what the animation that we looked at now, I want to use PyMol with you to look at this motor unit in detail. So and all you need for that right now is PyMol as itself. You don't need a file. So just open PyMol. So then give you a little bit of time. Actually, I would also like to know who is working with PyMol today, or who can work with PyMol today, so I can know how fast or slow I have to go. Because I'm sure I could do everything quite a lot faster if I don't have to wait. Okay, Yukiko, very nice. Somebody else who's using PyMol? Okay, Asta as well, nice. Okay, so today we are going to use a little bit different functionality of PyMol, which is actually using PyMol with two different files, right? So first we have to get the files that we want to use. Oh, nice. Koto's also using. Okay, to get those files, what you have to do is we're going to use a little bit of Python. I'm not sure I can call this using Python, but we're just going to use some commands. And one of them is called fetch, right? So what it does, it goes to a database that is online and gets a certain protein structure. So today, I noted on the presentation, we're going to get the structures 1DFK and 1DFL, right? So when you typed in fetch in the line at the bottom, you have space and then 1DFL, right? And then enter. Oops. What's that? Okay, one more time. Fetch 1DFL. Loading in. Okay. Okay, give me a second. I think I have to restart. Fetch 1DFL. Okay, all right. So this is what it's supposed to look like after you typed in the command and pressed enter. If you want to know what the computer did, you can also check at the top. Here it basically says what it did. I typed in fetch 1DFL and then it says, it gives me the title of the file that I loaded into the program, and then it tells me what it named it. This is all the information that you need. And then on the right, it basically appears here. Also important this time, unfortunately you don't see it, but in the drop-down menu from the top, if you click on display, I would also like you to click then on sequence, so you get the amino acid sequence of the whole protein. Right. Because what you can actually see here is that you have like 2 copies of the same protein. But we actually only want 1. Right? So to get only 1, it's a little bit annoying. There's probably a command, but I don't know it. So we have to select the amino acid sequence at the top and then delete it. Right? So first, what you can also see is there is some sequence that is not connected to the actual motor domain. So we also want to get rid of that one, so it's a little bit more clear. In case you don't find it, it's in the sequence after 835, I guess. It starts with chain B. This was what I showed you before in the presentation. This would be one of the light chains, but they don't actually interest us at the moment. Okay, one more time. How do you get the sequence? You have to press display at the top, at the very top, right, where it says File, Edit, Build, Movie, Display, and then click on Sequence. Just click once and then it should appear. Okay, so one more time, we want to select it, and then if you select it in the sequence, you can do right click and remove. Right? So what it does, it just removes the amino acid sequence. Right. So we're going to do the same with the sequence that we have here, which is chain C, which is another light subunit, which doesn't interest us. And then it looks like this. And then what's left is all the other part of the protein, right? The other exact copy. And we want to basically select all of this. Right, so you have to scroll to the right and then keep on selecting by just clicking and dragging. Select all of that, including the other light chains. Yeah, everything after that, yes. Okay. So if you ask me on Zoom, it probably will come out through YouTube as well. So probably typing is best for now. But yeah. Anyways. So then right click and remove. Okay, so what you're left with is only the chain A, and you see there are like some extra molecules. What you can do is actually you can just click on them and select them, and it would show you on the top which one that is, and you can again right click and remove. And also this one, and this one, right click, remove. Okay. So now we have only the motor domain of this protein. So if you go back to the presentation, well, that's not it. You have basically only the yellow and the blue part from this animation. Right? So some of the red of the yellow part we've deleted. But now we're basically left with the blue and part of the yellow. Looks like this. Oops, looks like this. Okay. All right. So now Actually, I'm going to give you some time. I guess most of you cannot do it as fast. So all you have left at the end is chain A from 1 to 835 amino acids, plus a magnesium, a vanadate, and an ADP, which is like in the binding site. So one thing to take a better look at the molecule, because now it rotates a little bit strangely, you can select the ADP because it's somewhat in the center of the protein. And what you can do is you can right click and then center, right? So it focuses the camera and the rotation on this molecule. And I also want you to focus a little bit on this point because this is where most of the magic happens, basically, in this protein. Right, so you can turn around, look at it from all kinds of sides. You can see there's like a little pocket, right, that is accessible for the ATP to bind. In this case, it's ADP, but they have, I think, the vanadate to block it and to make it look like it's a ATP for the protein. But yeah, so this what it would look like if ATP was bound, right? So little pocket, and if it's bound, basically the arm, I would just call it the arm, is in this kind of position. So next, we're going to fetch the other file, right? So we're going to type again in the command line at the bottom, fetch, then 1DFK. Okay, so what happens, you get a second protein on your screen, and in this case, well, it's actually almost exactly the same as the other one, right? So if you look at the top, somewhat, the sequence is very, very similar, right? So, but similar to the last one, we have some unnecessary parts, right? So we go towards the end again, we highlight it, and it's again the light chain that we don't want, and we right click it and we remove it. Same with the other light chain. And what you can see is in this one, you don't have an ADP. So logically, this is the state that the protein is in without the ADP. Right? So what we want to look at now, what is different? What changes in the structure between binding ATP and not having ATP bound? Right? So for now, well, that's kind of impossible because those two structures, they're not oriented correctly. So what we're going to do is a very simple command that is called alignment. So if you click on either one, it doesn't matter if you have 1DFL or 1DFK first. You can click on the A, and there's an option in the drop-down menu that says align. If you just hover over a line, you can see there are then more options. and one of them is to molecule, and that's what we want to do. We want to align to the molecule that's called 1DFK. Okay, and if you click it, basically this will happen. Okay, so now PyMol basically with calculations looked at, okay, at what regions with what amino acids could they actually overlap, and it's calculating a fit and puts them basically in a similar position. You will also notice that now you have the yellow lines that are kind of in between the two different proteins, right? What this kind of indicates is how different the position of these sequences are. So the more distance there is, the less well it is aligned to each other. Right. In this case, it's okay because actually there's a lot of movement in this protein. So you can actually see in the areas where the lines are longer, there's a lot of movement in between those two states. Right. But if you don't want to see them, you can also go to the right, where you now have the alignment of those two molecules, and you can click on hide and then CGO, right? And then the yellow lines are gone. Okay, so now we can see, if we look at the protein, actually let me get rid of the calcium, that there are two different states, right? So the arm is moving a lot, right? From right to left or left to right. So, and if you look at the pocket, right? Actually, let me center on the ADP again. Right click the ADP, center. Okay, so it's better to see. All that is different between those two proteins is this is bound or not bound. And what is happening in between those two states is a huge difference in conformation between the proteins, right? So it moves from one side to the others just because here there's this molecule bound. So it's a very, for me, it's a very interesting concept that even the smallest changes in binding to these molecules can have a huge effect on the overall structure. And that this change of the overall structure can be put to use in a machine-like fashion, right? So to go a little bit into the detail of what is changing, it's a little bit complicated in this case because in the one protein you might see here there is an end that is basically just unfinished. So this is a part of the model which could not be fitted well, which was left out, but actually it is somewhat important for the binding of the ADP and ATP because it would lie exactly over this pocket and would basically change how well the ADP can bind to the molecule. Anyways, if you go closer, you can see in this area, this small change basically propagates from with these loops and through this alpha helix, and is then amplified through even more alpha helices in the back, right? So this small change kind of gets bigger and bigger and bigger the further you go away. That's why it's also very interesting to look at it as kind of as a spring, because basically the molecule being there you could think of it as kind of pushing the spring together, and then when it goes away, the spring is released. Right? Plus the alpha helices, of course, look like springs, which kind of could contribute to the overall movement. So the most movement that you see, of course, is in this arm part, right? Which is, from its conformation, completely different. Okay, so I'm actually going to do something else, which you can't do at home because you don't have you only have a trial version of PyMol. One of the paid features of PyMol is also a morph command. So I'm going to type morph, then I'm going to give it a name. I'm going to call it transition. Then I'm going to say I want to morph 1dfl onto 1dfk. So it's going to take a little bit. So give it some time. I think last time I did it took about 30 seconds to a minute. So what the morph command does is basically it calculates how this protein could transition from one state into the other and makes a freeze frame of this transition, which can then be played as a movie. Right? So if you think about it, that takes a while to calculate. So if you were to do it, probably it would take longer, maybe. Depends on how good your computer is. Okay, now we actually have it. So what it automatically did, it hit all the other structures, and it only shows me a new part that it calculated that can move. And all you have to do to make it move is press the play button. Right? And you can see basically what is happening to the protein. Right? So it's huge movement that is happening, which all starts in this pocket, right? Which kind of opens up, and with this opening up, this kind of movement is exaggerated through these two helices. Right? So a lot of these bonds are reshuffling and changing their energy state to basically, like we talked about it, it's similar to the folding process, as that it switches from one stable conformation into another one. Like one is higher, one is lower on an energy level. Right? So you have to put in energy with the ATP to push it onto the other valley in the diagram that we looked at last time and a little bit today. Okay. So to get the full picture of what is happening, like in what I showed you in the beginning in the presentation, you would also have to load like 4 other states, which are, I think, not available. I'm sure. A lot of the things that you saw in the video, or not in the GIF, that I showed you in the presentation, is actually just theoretical. Right? So there are no actual models of these states. Right? The whole transition is kind of what people think and kind of calculated how this motor domain would move. Right? So overall, I think that's very, very interesting. And this is basically what your muscles do. This is all that's happening, right? So this protein, so if you want to move your arm, basically your brain gives this protein the signal, okay, now do this movement. Like push this arm from this direction in the other one. And then it pulls the actin together and makes it basically the distance of the cell smaller and creates force. All because of this little domain and the huge changes that it does when it binds ATP. Okay, so going back to the presentation, right? So now we kind of looked at what this switch is in conformation, right? Here it looks like a robotic arm, but we looked at what is actually happening on a molecular level. And I think because of what we talked about in the workshop so far, I think all of you are able to understand, and that's, I think, a great thing. Okay. So again, in summary, this is the whole function of the myosin. You have an ATP bound state. Let's just start at the bottom, where it does not like to connect to the actin. It changes its conformation, like we just saw. The arm moves around, which means those move. Then the ATP turns into ADP and P, which then allows the protein again to bind to the actin. ADP plus P is released, and through this release, basically the conformation changes again in the low energy state, I would say. And then when ATP binds again, it can detach and go into the high energy state again. Right? Okay. Maybe. Okay, right. We talked about this a little bit. There are many details and a lot of papers that talk about how this change happens and how this force is generated. And what we just looked at, the files I got from this paper, which talks about how this lever arm moves and how there's a converter attached to it. And I talked about the relay helix, which basically when the ATP binds, then exaggerates this movement and which brings the arm to turn around. Okay. So some of you might be thinking, okay, but how do I control this, right? So how does the body actually control when I want to make this protein change its conformation? Right? It's a very interesting and also very detailed process. So I don't want to talk too much about it. So what you see at the top here is basically a nerve ending, right? So this is basically something that would be attached from the nerve cells or from a nerve bundle onto a muscle cell, right? And maybe if you know how nerve cells work, all they do is they give a signal that then sodium concentration increases inside the cell through different transporters. This signal is then translated into a calcium signal, right? So calcium is actually the molecule that regulates if the myosin can bind to the actin. Because something I haven't talked about so far is it's not only actin, there's also something called troponin. You can see that here in purple, right? Troponin without calcium does not allow the myosin to bind to the actin. All right? But if it is activated because of the calcium increase, it can then bind, the myosin can then bind and do this power move, right? That we talked about. It can release this energy that it stored in a spring-like fashion from the ATP and can move the cell and make it contract. Right? And then again, the cell has to use ATP to restore the concentration to normal again, and then the myosin cannot bind anymore. It's interesting to mention that actually, well, the myosin can only move in one direction, right? So in theory, if you would only have one muscle, all it could do is contract, right? It could not relax by itself. So all that muscles do is basically contract and stay in this contracted state. The only way to relax the muscle and get it to zero again, like to the beginning state, is by actually having a muscle on the other side, right? Those are called agonist-antagonist movement. So for instance, in your arm, you have your biceps, but if you only had your biceps, you could only basically keep your arm in this, you can only move it up, but not back again, right? So to move it and relax your biceps again, you have to have your triceps to do the exact opposite movement, to bring the muscles into a state where they can then contract again. But in a way, this was only supposed to be a side note. Also, you can kind of see why, for instance, calcium is very important if you do a lot of sports, right? Because calcium is very important for the signaling and muscle activity. Also, muscles have a maximum frequency at which they can contract, which is given by, one, the counter movement, and also how well this this calcium concentration can be restored, because that takes a while, right? Compared to how fast you can give it a signal to contract, the thing that takes longer is actually to recover the calcium concentration. Again, to then be able to send the signal for the muscle to contract again. Okay. I hope you kind of understand how muscles work. If not, definitely ask me questions after the presentation today. I would be glad to explain a little bit more and talk about anything that's unclear. Otherwise, I would like to move to another molecule that is super, super interesting to look at, and that actually works very, very similar to myosin, to muscles. This one's called kinesin, right? You might know from the name comes from kinetic, like movement. And what it looks like is actually like it's walking. It's a protein that has somehow evolved to have a conformational change that looks like it's walking on the surface of another protein. So not only that, this is basically, it's transporting something also, right? So this is the main protein that is responsible for the transport of vesicles in cells. So all kinds of things are basically moved throughout the cell thanks to these proteins. All right, so in simplified form, this is what it looks like. So you have a change of one motor domain with the other one. So this conformational change is very, very similar to the muscle cell, or to myosin that we just looked at. Right? The swing of this arm through use of ATP is what makes this possible. So here's actually a very, very good movie. I couldn't explain it better. So I choose to just very quickly show you, which shows how this walking type movement is facilitated. I hope the quality is okay. The movie the movie quality was kind of bad before. Oops. Nope.
Two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
Okay, very well. I think that's super interesting. It's so you see there's a lot of similarity between myosin, but it's actually not exactly the same. Like for one, it binds not actin, but microtubules, which are, well, a molecule called tubulin. Also, it's kind of like the inverted process, because here you can see that actually the ATP binding state is the one where it binds to the microtubule. Whereas in comparison with the muscle or the myosin, if myosin binds ATP, it actually releases itself, right? And then switches the conformation. Here's the other way around. So actually it changes the conformation with ATP and P. Okay, but in itself, it's a very, very similar process. And they are related proteins, right? So what it looks like, it's actually kinesin can only move in one direction. So if you look at the microtubule, there's something called a plus and a minus end. The plus end is directed towards the outside of the cell, which means the plasma membrane, and the minus end is directed usually towards the cell nucleus. So the kinesin only transfers things away from the nucleus, and there's another protein called dynein, which well, it's a little bit a different movement process, but it moves it in the different direction, right? So basically, actually, I didn't want to show you dynein because it's a little bit more complicated, but kinesin is like if you were to put your wrists together and then the two hands like walking over each other, but dynein is actually like two different legs which slide along the microtubule to move in the other direction. It's a super interesting process, which I wanted to show you. And it's if you would compare it, for instance, to a combustion engine, to a motor, right? So it's a lot smaller, of course. It uses ATP instead of gas. But interesting to note, it's actually 3 times as fast as an average combustion engine, right? And it also has a much, much higher efficiency. So if we were to design an interesting transportation device with high efficiency, maybe we could take a look at the kinesin. Okay. So what it looks like in the cell, because now with our technology, of course, we have ways to not only look at it as a simulation or as a 3D model or whatever, we can actually look at it in live cells, right? So what you can see on the left is in green are microtubules, and the red is basically the kinesin moving along them. Or here's another example. Here you can actually see the movement in 2 directions. Or something that looks more like firework, where the microtubules are all originate in one single bundle and then spread out. And this is the transport that is happening along them. So this is used for all types of cell functions, for moving things inside the cell. Even the nucleus can be moved along microtubules with the kinesin molecules, right? Because inside cells, there's always movement, right? Everything is movement. And if you have ever looked at even a simple leaf cell, you can see there are many, many things that are, they look alive, right? They're not static as you would see them in your textbooks. And a lot of this movement is from those kinesin molecules, kinesin dynein. Also, another interesting thing that I wanted to show you is, if you think about squids, right? Squids have a very interesting adaptation, right? They can change color, as you might know. And they can change color because of specific cells that you can see on the left in the picture. These cells are basically black spots that are getting smaller and bigger. These spots are controlled by hormones in the squid, right? And the hormones, like for instance adrenaline, can tell them to get bigger or smaller. So, for instance, when a predator comes, the squid gets scared. It will excrete adrenaline. This adrenaline will then cause the spots to get bigger, and therefore the squid looks dark and black and menacing. If you think of it on a molecular level, I want you to take a look at the video. Right, so these are basically similar to the black spots that you see. So what is happening in this case, the dynein that we just talked about is moving vesicles full of pigment, right, of color, and is moving them towards the inside of the cell into a single spot, right? Therefore concentrating the color effect and making the black spots smaller. Right? And to spread them out again, basically all these things are then facilitated by kinesin again, with a different hormone probably, or even the lack of adrenaline will make them spread out. I'm not sure exactly. Okay, and with this, we're at the end of today's workshop. Yes, so we talked about how muscles work, and we talked about another form of movement in cells, which is performed through kinesin. Okay, for any questions or Otherwise, comments, please join me on Zoom after the live stream goes off. And otherwise, I will see you guys next week. Thanks for coming.